Welcome, curious minds, to another deep dive into the astonishing, often invisible corners of our world. Imagine this bustling metropolis just teeming with life, constantly interacting, changing, but it's completely hidden from view. That's kind of the reality of the microscopic universe living inside our very bodies, you know?
Yeah.
A realm full of incredible organisms, and we often don't even know they're there. Today, we're not just dipping our toes in. We're really plunging headfirst into this hidden world. We're going to focus on two really intriguing protozoan parasites, Entamoeba and Leishmania. Our mission, basically, is to unravel what makes them tick, understand their impact on us, on human health, and maybe most surprisingly, uncover some, unexpected connections, especially when it comes to dentistry. It's shaping up to be a pretty eye-opening exploration into how these tiny single-celled things exist, how they interact with us, and why knowing about them is crucial even in fields you might not immediately think of.
Exactly. And this deep dive, the aim is really to give you a comprehensive but still accessible understanding of these microorganisms. They often get overlooked. We've carefully pulled together insights from recent articles, some cutting-edge research, and sort of distilled it down. Think of it like a shortcut. A way to get well informed about these vital players in the microbial world. And really shows why studying them isn't just for infectious disease specialists. It's for anyone wanting to grasp how connected our health really is, from our gut right up to our gums.
Okay, let's unpack this realm of tiny living things then. To start, let's just get the basics down. When we say protozoa, what exactly are we talking about? How do they fit into the bigger picture of microbes?
That's a great place to start. Yeah, the term protozoa can sometimes feel a bit vague. But at their core, protozoa are unicellular eukaryotic microorganisms. Now, breaking that down a bit, unicellular just means they're single-celled, like bacteria. Pretty simple. But the really key difference is eukaryotic. See, bacteria are prokaryotes. They don't have a true nucleus or those other membrane-bound bits inside. Eukaryotic cells, like protozoa, are much more complex. They have proper nucleus for their DNA, plus other specialized compartments like mitochondria.
Okay, so definitely distinct from bacteria, a different kind of microbial life, maybe more structurally complex, even though they're still just one cell.
Precisely. They're both microorganisms, sure, but protozoa are on a different branch of the evolutionary tree. You can maybe think of it like a simple studio apartment versus a multi-room house. Both are single dwellings, right? That's kind of the difference. Bacteria are functionally simpler. Protozoa have more internal organization. And within that big group of protozoa, their lifestyles, their roles, they vary wildly. Some are totally harmless residents living alongside us, maybe even in us, causing no trouble at all, like silent roommates.
Yeah.
But others are serious pathogens, capable of causing really significant diseases, even life-threatening ones. It's a remarkably diverse group, and understanding the diversity is absolutely key.
That sets the stage perfectly. So let's dive into our first specific protozoan, Entamoeba. What do these things actually look like? The sources describe them as large motile amoeba. Can you help us visualize that? Especially since we're talking microscopic scale here.
Certainly. And large is always relative in the micro world, isn't it?
Yeah.
But it gives us a clue. So when the sources say large motile amoebae, let's take Entamoeba gingivalis as an example, one we'll talk more about later. It's typically around 12 micrometers in diameter. Now, to put that in perspective, your average human red blood cell is maybe 6 to 8 micrometers across.
Okay, so E. gingivalis is roughly double the size of a red blood cell. Still tiny, obviously, but bigger than I might have pictured.
Exactly. Still totally invisible without a microscope, but pretty substantial for a single cell compared to, say, a typical bacterium, which might only be 1 or 2 micrometers long.
That helps a lot with the scale. And motile amoebae, what does that tell us about how they move or their structure? Do they have a set shape?
Right. Motile amoebae points to their really characteristic and honestly quite fascinating way of moving. They don't have little hairs, cilia, or tails, flagella like some of the protozoa. Instead, they move by extending these temporary bits of themselves called pseudopods. Literally false feet.
False feet.
Yeah. Imagine like a blob of jelly continually pushing out parts of itself, anchoring that part, and sort of flowing the rest of the body along. They essentially ooze or crawl. Their shape is constantly changing, quite amorphous. And this movement isn't just for getting around. It also lets them engulf food particles or even host cells using a process called phagocytosis. So you've got this relatively large single cell that's actively shape-shifting, moving, extending these false feet to explore and eat. It's pretty dynamic for something so small.
Fascinating, like tiny shape-shifting blobs moving with purpose. Okay, let's zoom in on the more infamous member of the Entamoeba family now, the one that causes significant human illness. Entamoeba histolytica, it really is the star pathogen of the group, isn't it?
Oh, absolutely. Entamoeba histolytica is, without a doubt, the most well-known and clinically important pathogen in the whole Entamoeba genus. It's the culprit behind a condition called amoebiasis. When it gets severe, it's often specifically called amoebic dysentery. And how many people usually get it? Well, it's pretty common in many parts of the world, typically through ingesting food or water contaminated with the organism's cysts.
Cysts, like little protective shells.
Exactly. These cysts are incredibly tough. They're the dormant protective form of the parasite. They have this strong wall that lets them survive outside the body, things like stomach acid, temperature changes, even chlorine to some extent. So someone swallows them, they pass through the stomach, the small intestine, and then they reach the large intestine. And there the conditions are just right for them to exist, basically transform into their active mobile form. That's called the trophozoite. And it's these trophozoites that actually cause the disease.
Okay, so contaminated food or water, the dormant cysts wake up in the gut, and then these active trophozoites are let loose. What happens then? Is it always a severe illness, or does it vary?
That's a really excellent question, and crucial because it highlights the huge spectrum of disease Entamoeba histolytica can cause. It's definitely not one size fits all, which makes it complicated, from a public health standpoint. Symptoms can range from completely asymptomatic, someone carries the parasite, might even shed cysts, but feels totally fine, all the way to very severe, even fulminant colitis.
Fulminant? That sounds bad.
It is. It means extremely aggressive, rapidly worsening inflammation of the colon. It can be life-threatening if it's not treated quickly and effectively. So you can have silent carriers potentially spreading it unknowingly, or someone suffering from an acute debilitating form. Quite a range.
Wow, silent to severe. When it does get severe, like with dysentery, what's actually going on inside the body? How does this tiny amoeba cause so much damage to the intestinal wall?
All right, when we talk dysentery, we usually mean an infection caused by what we call an enteroinvasive microorganism. This means they don't just hang out in the gut. They actively invade the intestinal lining, the mucosa. And Entamoeba histolytica is a classic example. Those active trophozoites, the amoeboid forms, they have specific enzymes like proteases. These enzymes let them literally break down and degrade our tissues.
Oh, wow.
Yeah, they latch onto the intestinal lining, secrete these enzymes, and create these little areas of necrosis tissue death. Then they invade the intestinal wall, burrowing deeper. This leads to significant inflammation, the formation of these characteristic flask-shaped ulcers you can sometimes see, and just widespread tissue destruction. It's this direct attack and invasion that causes those severe symptoms we associate with amoebic dysentery.
And what are those specific clinical signs? If someone has this invasive form, what would they or their doctor typically notice?
The signs are pretty distinctive, yeah, and they directly reflect that invasion we just talked about. The most obvious symptom is frequent, often urgent, watery stools. And what's really characteristic is that these stools very often contain visible blood and mucus. That helps distinguish it from some other types of diarrhea.
Okay, blood and mucus, that's a key sign.
Definitely. Patients also typically have significant abdominal pain. Could be cramping, could be severe discomfort, often down in the lower abdomen. Another really distressing symptom, a hallmark of this kind of inflammation, is tenesmus.
Tenesmus.
Yeah, it's that painful but ineffective straining when you try to defecate. You feel this urgent need to go, you strain hard, but little or nothing comes out, and you still feel like you haven't finished. It's very uncomfortable.
That sounds awful.
It is. Fever is also common, showing there's a systemic inflammation going on as the body tries to fight back. And of course, with all those frequent watery stools, dehydration can become a really serious problem really quickly, especially dangerous for kids or the elderly. All these symptoms are direct results of the parasite damaging that intestinal lining.
Sounds incredibly unpleasant and potentially dangerous, especially the dehydration risk. So if someone shows up with these symptoms, how do doctors figure out it's actually Entamoeba histolytica and not something else causing gastrointestinal trouble? What's the lab diagnosis like?
Well, the primary, most direct way to diagnose Amoeba histolytica is by examining stool samples. A skilled lab tech will usually prepare what's called a wet film of concentrated feces. Basically, they take a tiny bit of stool, mix it with a drop of saline on a microscope slide, put a cover slip on top. The saline helps keep any active trophozoites alive and moving for a bit, so you can actually see their characteristic movement, those pseudopods we talked about.
Oh, so you can literally see them moving.
Sometimes, yes. It's quite definitive if you see them. They also often use iodine mounts. Iodine stains the internal structures, the nucleus, other bits inside the cysts and trophozoites, making them much easier to see and identify properly. So under the microscope, the technician is hunting for those active trophozoites, the mobile feeding stage, and also the tougher infectious cysts. They'll also look for other things, like worm eggs, just in case there are co-infections. And seeing red blood cells or pus cells in the stool also supports the diagnosis of an invasive infection.
So spotting the actual organism or its forms in the stool is key. Are there other tests, maybe for cases that are less clear-cut or chronic?
Yes, absolutely. Direct microscopy is the front line, especially for acute cases, but it's not the only tool. For a fuller picture, particularly if the infection might be chronic or maybe less symptomatic, or if there's a suspicion it's spread beyond the gut, like to the liver, which can happen.
It can spread to the liver.
It can, yeah. Extraintestinal amoebiasis. In those cases, serological tests are really useful. These are blood tests, things like the indirect hand agglutination assay, IHA, or ELISA tests. They don't look for the parasite itself, but for the body's immune response-specific antibodies in the patient's serum. Finding these antibodies suggests a past or current infection. A positive result, especially if you also see trophozoites in the stool, strongly points towards active invasion. So it's often a combination approach. Direct visualization plus looking for the immune response helps ensure accuracy.
That makes total sense. Look for the bug itself and the body's reaction to it. Okay, so once it's diagnosed, what's the usual treatment for Entamoeba histolytica causing amoebobiasis?
For symptomatic or invasive Entamoeba histolytica, the standard drug, the drug of choice, is metronidazole. It's a very effective antibiotic, works really well against anaerobic parasites and bacteria too. It basically messes with the parasite's DNA and protein production, killing the active trophozoites. Sometimes, after the metronidazole, another drug, maybe something like paramomycin, is given. This is a luminal amoebicide. It works inside the gut lumen to kill off any remaining cysts, helping prevent a relapse or stop the person from spreading it further, especially if they were an asymptomatic carrier.
Okay, got it. Metronidazole is the key player there. All right, we've really dug into Entamoeba histolytica, the gut invader. But now, here's where it gets really interesting for our dental implications chat. Let's shift focus to Entamoeba gingivalis. This one is found specifically in the mouth, in the oral cavity. How common is it there, and what do we know about its connection to oral health?
Right, Entamoeba gingivalis. It's actually quite prevalent in the mouth. Our sources say it's frequently found in plaque samples, and there's a strong association. You find it much more often in people with poor oral hygiene and those who already have periodontal disease or gum disease. The worse the hygiene, the more advanced the gum disease, the higher the chance of finding E. gingivalis. This correlation strongly suggests it likes a certain kind of oral environment.
So it hangs out in unhealthy mouths, but what's its actual role? Our sources mention its precise role in oral disease is still unclear, like it's just a bystander, a mere passenger. That sounds like a fascinating puzzle, doesn't it? If it's so common in unhealthy mouths, why isn't it considered a definite cause?
That's exactly the debate, the sort of paradox around Entamoeba gingivalis has been going on for a long time. Despite being so common in diseased mouths, it's generally classified as a harmless saprophyte or, a mere passenger. What that really implies is that it doesn't seem to initiate the oral disease itself. Instead, it seems to thrive opportunistically. It benefits from the conditions created by other things, mainly poor hygiene and existing inflammation.
So it takes advantage of the situation.
Exactly. Think about it. If someone has poor oral hygiene, they'll have lots of accumulated food debris, dead cells, tons of bacteria, especially anaerobic bacteria, hiding in those periodontal pockets between the teeth and gums. This creates a really nutrient-rich, low-oxygen environment. E. gingivalis just loves that. It basically moves in and feeds on the bacteria and the cellular debris. It proliferates where there's plenty of food and not enough cleaning. So its presence is more like an indicator of an unhealthy mouth, rather than the direct cause of the problem. It's more consequence than cause.
That's a really important distinction, not causing the issue, but benefiting from it. But are there any intriguing clinical observations about it? Anything that maybe hints at something more, even if it's not officially a pathogen?
There are definitely some interesting observations that keep researchers curious, even if they don't really change its overall classification for now. For instance, it's been noted in higher numbers in periodontal tissues of patients undergoing radiotherapy, like for head and neck cancers. Radiotherapy can really mess with the oral tissues, cause mucositis, change the microbial balance, maybe creating an even better environment for E. gingivalis.
Okay, that makes sense.
And perhaps even more interestingly, it's been observed in people who are taking metronidazole.
The same drug used for Entamoeba histolytica.
Exactly. The same drug we just talked about for the pathogenic gut amoeba. So finding E. gingivalis in patients who are already on this specific amoeba-targeting drug, well, It creates a curious link, doesn't it? It's almost like seeing a distant relative react to the same medicine. It hints at maybe shared biology or sensitivities, even if its role in the mouth is officially considered harmless from a disease perspective. It's one of those details that makes scientists go, and keep asking questions.
It really does make you wonder if there's more to the story. But if it's hard to pin down its exact role, what are the challenges in actually studying E. gingivalis? Why is it so difficult?
That's one of the biggest hurdles, absolutely. While you can easily spot it under a microscope in a plaque sample, you can see it moving in a wet mount. The major challenge is that Entamoeba gingivalis is incredibly difficult to culture in the lab.
Can't grow it easily.
Nope. It has really strict anaerobic requirements. It hates oxygen. And it needs a very complex medium to grow, often needing specific bacteria or even cell cultures mixed in to provide the right nutrients. This inability to easily grow it in isolation really hampers research into its potential pathogenicity. I mean, without being able to reliably grow it in a controlled lab setting, it's super hard to do experiments that could definitively prove or disprove whether it actually causes or worsens oral disease. Researchers are mostly limited to observational studies, seeing where it shows up. And that shows correlation, sure, but not necessarily causation. That's why the harmless passenger debate kind of continues.
Okay, so understanding these characteristics and challenges, what does this all mean for dentists, for oral health professionals? If Entamoeba gingivalis is mainly just a passenger, does it need any specific dental treatment?
This is a really key takeaway from our sources, and important to clarify. For Entamoeba gingivalis, even though it's strongly linked with poor hygiene and gum disease, its actual pathogenic role isn't well established. It's primarily seen as a commensal, or a saprophyte just benefiting from the unhealthy conditions. Therefore, our sources don't indicate that E. gingivalis itself causes oral disease or needs specific dental intervention beyond general improvements in oral hygiene.
So focus on cleaning up the environment it likes.
Exactly. The focus for dentists stays on tackling the underlying problems. Meticulous plaque control, professional cleanings like scaling and root planing to remove calculus tartar, and really working with the patient to improve their daily brushing and flossing habits. These actions naturally reduce that opportunistic environment, the food debris, the anaerobic conditions that E. gingivalis thrives in. So finding E. gingivalis is more like a biological red flag signaling an unhealthy mouth, rather than something that needs its own targeted antibiotic treatment. Presence doesn't always equal causation or the need for specific therapy.
That really clarifies the situation for E. gingivalis in the dental chair. A great example of how understanding the microbes in one spot connects to the bigger health picture. But what about the serious systemic one, Entamoeba histolytica? It doesn't cause direct oral symptoms, but how do those general principles of managing systemic diseases apply in dentistry?
Right. For systemic Entamoeba histolytica infections, the dental implications fall under those broader, really fundamental principles of managing patients with any kind of systemic disease. It's all about taking a holistic view of the patient, even if the main infection isn't showing up directly in the mouth or posing a direct transmission risk in the dental setting itself. And this brings us back to a really fundamental point. Dental professionals absolutely need a solid grounding in general and medical microbiology. This isn't just academic stuff. It's crucial for them to accurately diagnose oral infections that might actually be linked to a patient's overall systemic health or their treatment plan.
Like connecting the dots.
Exactly. For instance, if a patient has active severe amoebic dysentery, they might be dehydrated, maybe their immune system is stressed, they might just be generally unwell. Now, the dentist isn't treating the amoebiasis, obviously. But knowing about it means they might need to postpone elective procedures, be prepared for potential issues like slower healing due to the patient's compromised health, or maybe coordinate care with the patient's physician. This foundational knowledge lets them see the whole patient, not just the teeth and gums in isolation.
So less about treating the amoeba in the mouth, much more about knowing the patient's overall health context, and crucially, ensuring a safe environment for everyone.
Precisely. And that leads straight into the critical importance of standard infection control precautions, SICPs, in the dental clinic. These protocols are absolutely non-negotiable. They're the bedrock of safety, and they operate under one vital principle. Assume every patient is potentially carrying an infectious disease.
Universal precautions, basically.
Exactly. Universal precautions. Every patient, no matter their known health status, is treated as if they could potentially transmit something, virus, bacteria, protozoa, whatever. This minimizes risks, even from systemic conditions like E. histolytica that aren't typically transmitted orally in a dental setting. It protects the patient from cross-contamination, and it protects the whole dental team. And it's really important to stress again, Our sources don't specifically mention oral manifestations or direct transmission risks in the dental clinic for Entamoeba histolytica. This is different from some other systemic infections, like, say, syphilis, which can cause oral sores, or viral hepatitis, which carries specific bloodborne risks. For E. histolytica, the implication is more about that comprehensive patient management, understanding their health status, and rigorously applying those universal infection control standards, rather than worrying about specific oral symptoms or direct dental transmission pathways needing special procedures. It just ensures care happens safely within the patient's broader health context.
Okay, fantastic. We've really explored the Entamoeba world, the gut invader, the oral passenger, and the dental connections. Now, speaking of complex microbial interactions, our next protozoan, Leishmania, offers a totally different and equally challenging perspective on how parasites and hosts interact. Let's shift gears now and focus on this intriguing organism.
It certainly does offer a different perspective. Leishmania species are also protozoan parasites, just like Entamoeba. But their whole lifestyle, how they're transmitted, the diseases they cause, is quite distinct. While Entamoeba is mainly about the gut or opportunistically the mouth, Leishmania has a much more systemic presence. It interacts very directly, very intimately with the body's immune cells, often circulates in the bloodstream, and humans get it through the bite of infected female sandflies. The bite introduces the parasite into the skin.
Sandflies. Okay, so an insect vector is involved here, unlike with Entamoeba.
Correct. That's a key difference in transmission.
And once they're inside us, how do doctors even see these organisms? Are they also in stool samples, or is identification different because they're systemic?
The main way to observe Leishmania is quite different from Entamoeba, yes. Our sources indicate they can be seen microscopically in stained peripheral blood films. That immediately tells you they're in the bloodstream or tissues, not primarily the gut or mouth. Big difference from E. histolytica in school or E. gingivalis in plaque. So a blood sample is taken, spread thinly on a slide, stained with a special dye like James's stain, and then looked at under a powerful microscope. The parasites, in what's called their amastigoot form, appear as these small oval bodies usually found inside host cells, especially macrophages.
Inside macrophages. Wow. And what's really fascinating here, you mentioned, is how Leishmania interacts with our defenses. It's not just hanging out. It's engaging in this really complex dance with our immune system, specifically those macrophages. Can you elaborate on that? It sounds like a real biological battle.
That's truly the core of how Leishmania causes disease, and yes, it is a sophisticated battle. Like some other major protozoan parasites, think Plasmodium causing malaria or Trypanosoma causing sleeping sickness, Leishmania species have this profound, often manipulative effect on our immune cells. They specifically target and get inside macrophages. Remember, macrophages are normally our frontline defenders, the cells meant to engulf and digest invaders.
Right, the cleanup crew.
Kinda, yeah. But once Leishmania infects these macrophages, it actually stimulates them to release a powerful flood of inflammatory cytokines. These cytokines are like tiny chemical messengers, the body's alarm signals. Really potent molecules like interleukin-1, IL-1, interleukin-6, IL-6, and tumor necrosis factor alpha, TNFC. These aren't just jargon, they're key drivers of the inflammation, the fever, the immune responses you see in these infections. It's part of the body trying to fight back, but paradoxically, Leishmania uses the very cells designed to kill it as its hiding place.
So they hijack the body's own defenders, turning them into, well, participants in the disease. But if macrophages are supposed to destroy invaders, how does Leishmania manage to survive inside them? That seems to completely defy their purpose.
That is the remarkable and frankly quite clever part of their strategy, surviving and even replicating within the macrophage. Instead of getting digested by the macrophage's powerful enzymes and the acidic environment inside, Leishmania has evolved these really sophisticated ways to resist. They can, for example, stop the part of the macrophage holding them, the phagosome, from fusing with the digestive parts, the lysosomes. They can also neutralize some of the toxic chemicals like reactive oxygen species that macrophages produce to kill things.
Wow, they have counter defenses.
They really do.
Yeah.
It effectively turns that host defense cell into a sanctuary, a safe haven. They can hide there, replicate, shielded from other immune attacks that might target pathogens floating freely outside cells. This intracellular survival is a key trick for many successful parasites. It lets them persist. But the immune system isn't totally powerless. Our bodies have counter moves too. The outcome of the infection often hinges on this delicate balance. If macrophages get properly activated, usually by specific immune cells called Th1 cells, a type of T helper cell, these supercharged macrophages can overcome Leishmania's tricks and kill the parasites. So it really illustrates this complex, ongoing fight, a dynamic struggle. The type of disease someone gets, maybe just skin sores, maybe severe systemic illness, reflects this immune tug of war.
So not a one-sided fight. The immune system pushes back. And I think I read that leishmaniasis can also cause a kind of hypersensitivity. What does that mean here? Is it like an allergy?
That's a good question. Hypersensitivity can be tricky. In this context, leishmaniasis is noted as one parasitic infection that can lead to immune complex-mediated hypersensitivity. It's not usually like an immediate allergy like hay fever. Instead, it's more about the body's own immune response, sometimes going a bit haywire. In trying to clear the parasite, the immune system can form these things called immune complexes. These are basically clumps of antibodies stuck to bits of the parasite.
Okay.
If too many of these complexes form, or the body can't clear them out efficiently, they can get deposited in various tissues, maybe the kidneys, joints, blood vessel walls.
Yeah.
And once they're stuck there, these complexes can trigger more inflammation, activating other parts of the immune system, like complement. This prolonged or misplaced inflammation can then lead to tissue damage and contribute to the disease symptoms, sometimes far from where the parasite initially was. So it's an example of the immune response, while trying to help, inadvertently causing some collateral damage.
Okay, that makes sense. It's the response itself causing problems. So given this complex immune interaction and its presence in the blood, how is leishmaniasis diagnosed in the lab? Similar to Entamoeba or different methods?
The main diagnostic method is that microscopic examination of stained peripheral blood films we mentioned earlier. But also smears taken from tissue biopsies can be crucial, especially from skin lesions if it's cutaneous leishmaniasis, or maybe bone marrow if it's the severe visceral form. For blood, they use both thin and thick smears. A thin smear lets you see individual blood cells clearly, check the parasite's morphology inside the cells, helps identify the species. A thick smear uses a much thicker layer of blood, and they lease the red blood cells. This concentrates everything else, making it much easier to spot parasites, even if there aren't many of them.
So different techniques for different goals?
Exactly. It's more like how you'd look for malaria parasites in blood, rather than looking in stool like for E. histolytica. And nowadays, molecular methods like PCR are also used more and more. They're very sensitive and specific.
And treatment. Once identified, what does treatment look like? Are specific drugs mentioned in our sources for Leishmania?
This is an important point to clarify based on our material. The sources we reviewed don't actually specify particular treatments or drug regimens for Leishmania. They just note that this information wasn't provided in the scope of this deep dive. However, they do make a very crucial general point relevant to our dental discussion. Anti-protozoal agents, as a whole class of drugs, are typically not prescribed in dentistry. This really highlights the different roles of medicine and dentistry. A patient might be getting systemic treatment for leishmaniasis from their doctor, but prescribing those kinds of drugs isn't usually part of dental practice.
That's a very clear and important distinction. Okay, so no direct oral signs, no specific dental drugs prescribed by the dentist. But if there are no direct dental implications for Leishmania, why do dental professionals still need to know about it? What's the broader connection here between a systemic parasite and oral care?
This is where those indirect implications become so important. And it really loops back to our main theme of holistic patient care. Our sources are clear. No direct oral manifestations described, no specific dental issues caused by Leishmania itself. Unlike, say, oral thrush, which you can see in the mouth. However, just the fact that a patient has a systemic parasitic infection like leishmaniasis, or really any chronic systemic condition, it fundamentally changes the context for their dental care. Firstly, it comes back to that vital recurring point. Dental students and dentists need that basic but solid understanding of general and medical microbiology. It's not just about memorizing bug names, it's about being able to diagnose oral infections that could be linked to the patient's overall health. Think about a patient with chronic leishmaniasis. Their general health might be poor, their immune system might be altered from fighting the parasite constantly, maybe they're on complex drugs. All these things can impact their oral health and how they respond to dental treatment. Maybe they're more prone to common mouth infections. Maybe healing after an extraction is slower. Maybe they react differently to anesthesia. Understanding that systemic background is vital for planning safe and effective dental work.
Right. It's about understanding the whole person sitting in the chair, not just their mouth in isolation. And how does knowing about Leishmania's unique immune interactions, that complex dance you described, help a dentist?
Excellent question. It provides really deep context. Understanding how Leishmania messes with the immune system inducing cytokines, hiding in macrophages, causing hypersensitivity, helps the dental professional appreciate the systemic health situation more deeply. It gives insight into why a patient might have general fatigue or a compromised immune response or systemic inflammation. Again, the dentist isn't treating the leishmaniasis, But having that holistic view is crucial for providing comprehensive care, planning treatments well, anticipating risks. It helps piece together the patient's health puzzle. For example, if their immune system is busy fighting Leishmania, maybe their ability to heal from gum surgery is reduced. Or they're more susceptible to opportunistic oral infections. It's seeing the patient as this interconnected system. And this leads to the third and maybe most universally critical indirect implication: infection control. Again, no matter what specific infection a patient might have, known or unknown, dentists must rigorously apply all aspects of infection control. Standard infection control precautions, SICPs, operate under that fundamental principle. Treat every patient as potentially infectious. This universal approach manages risks, even from systemic conditions like leishmaniasis that aren't typically spread in a dental setting. It protects everyone, patients, and the team.
So, considering all this, what does it really mean for you, the listener, maybe as a patient, or perhaps someone thinking about a career in dentistry? How does this deep dive into microscopic life actually translate into safer, better healthcare?
It means that dental professionals are much more than just technicians fixing teeth. They are absolutely vital members of the overall healthcare team. They're expected not just to understand these complex microbiological ideas, but also to advise their own teams, and really importantly, to help calm patient fears about infection transmission. Their knowledge of microbiology and infection control isn't just for diagnosis or planning, it's also a crucial public health communication tool. They act as trusted sources of information, helping keep the clinical environment safe, calm, and informed for everyone. It really underscores the responsibility of being a healthcare provider, a responsibility that goes way beyond just the specific body part they focus on. It's about building trust and ensuring safety at every level.
What an incredible journey today. We really untangled the formidable Entamoeba histolytica, a true gut invader, cause of serious dysentery. Then we looked at its curious relative, Entamoeba gingivalis, that enigmatic passenger in the mouth, whose story seems mostly about being a harmless scavenger, but whose presence definitely signals an unhealthy environment.
And then we completely shifted gears to Leishmania, that systemic parasite engineering the remarkable, often debilitating dance with our immune cells, especially macrophages. We saw the key differences, right? Gut versus mouth versus bloodstream, established pathogen versus passenger. But there are common threads too. They're all fascinating protozoa, all microscopic, and all carry important implications, sometimes indirect for general health and, yeah, surprisingly, for dental awareness.
And the so what here, the main message, feels really clear and profound. It's just how crucial a broad understanding of microbiology is for all healthcare professionals, including dentists. Imagine a patient comes in maybe with unexplained fatigue and also chronic gum inflammation. Without that wider understanding, a dentist might miss subtle clues connecting those oral signs to something deeper, something systemic, even something seemingly remote like a parasitic infection. It's about building that comprehensive picture of health, realizing it's all connected from deep in our gut right up to our mouth.
Absolutely. Even these indirect links to systemic infections really hammer home the importance of universal precautions and truly holistic patient care. It's about recognizing every patient as this unique biological landscape. And the more we know about the microbial world, the better equipped we are to navigate that landscape safely and effectively. It's a constant learning process that benefits everyone involved.
Which leaves us with a question for you to ponder long after this deep dive finishes. How does our growing, more nuanced awareness of these seemingly distant microscopic threats, these tiny lives sharing our existence, how does that actually contribute to a safer, more informed approach to our own daily health habits and the medical care we seek and receive? And what does it really mean to grasp that continuous unseen dance between microbes and our immune systems, a dance happening right now inside all of us every single moment? We really hope this deep dive gave you a fresh perspective on that hidden world inside us and the profound connections it has to our health. Thank you so much for joining us on this exploration. Until next time, keep that curiosity sparked and keep digging beneath the surface.